Let's discuss on tryptophan metabolism. Tryptophan is an amino acid which is belong to category of heterocyclic amino acids. It is the only amino acid which contains a specialized ring, that is the indole ring. So this is the structure of tryptophan. This is the general skeleton of all amino acid. This is CH3COOH, and the side chain R group is this indole ring, and its name is chemical name is alpha amino beta indole propionic acid. It is an essential amino acid and is both glucogenic and ketogenic. Following products can be derived from the tryptophan. They are alanine, acetoacetyl coenzyme A, formyl group, NAD and NADP, which are the coenzyme of niacin, serotonin, melatonin, hydroxyindole acetic acid as an excretory product, then endicane, that is also an excretory product. And as far as its metabolism is concerned, so it is being metabolized through two pathways. One is the anthraquinone anthranilate pathway, and second one is the serotonin pathway. So let's see the kynurenine anthranilate metabolic pathway of tryptophan, which is important for production of so many biologically important products like alanine and niacin, acetyl coenzyme A, etc. So tryptophan in the first reaction is converted up into the N-formyl kynurenine. The first reaction of usually most of the amino acid used to be transaminase, but tryptophan does not undergo the transamination reaction. So via the enzyme tryptophan pyrrolase, it converts to N-formyl kynurenine with the loss or breakdown of this pyrrole ring. And further, formamidase enzyme will lead to release of one carbon species, that is a formate, and which will be further utilized in one carbon pool. So then further this N-formyl kynurenine is converted up into the kynurenine, which is responsible for production of three important products. Kynurenate itself can be converted to the kynurenate, alanine, and the anthranilate. So we'll see further how they are being synthesized. Then kynurenine, in the presence of kynurenine hydroxylase, there is a hydroxylation of kynurenine in one step pathway, and this is the 3-hydroxy kynurenine. The enzyme is kynurenine hydroxylase. It requires the molecular presence of oxygen and NADPH. So further, this 3-hydroxy kynurenine can be converted into xanthanurate, and the loss of ammonia. Further, the 3-hydroxy kynurenine converts up into the 3-hydroxy anthranilate, while the enzyme kynurenase, which requires the presence of pyridoxal phosphate, that is the active form of vitamin B6. Without this or in deficiency of this PLP, there is no production of further products. And here at this step, the pathway is diverted to the synthesis of huge quantities of xanthanurate, which can be excreted in the urine. So in this particular reaction also, there is a production of alanine, another important biologically important product, that is a non-essential amino acid which is glucogenic in nature, which can further be converted up into the glucose via gluconeogenesis. Further, 3-hydroxyanthranilate in the presence of enzyme 3-hydroxyanthranilate oxidase. So it converts up into the 2-amino 3-carboxy muconate semialdehyde, which further by the enzyme piconylate carboxylates converts up into the piconylate. So 2-amino 3-carboxy muconate semialdehyde can be spontaneously converted up into the quinolate nucleus with the loss of water molecule. So there is a recyclization of uncyclized molecule. Then this quinolate further via the PRPP pathway, it can lead to the synthesis of ribose 5-phosphate nicotinate mononucleotide, which is NAD and responsible for the synthesis of NADP. So this is how the production of two very, very important biologically important product, NAD and NADP, which are the active form of vitamin B3. Then further, this muconate semi-aldehyde can also be converted up into 2-amino muconate semi-aldehyde with the loss of carbon dioxide, which can further, after several steps, it can be converted up into the glucoteryl coenzyme A, which will be finally converted up into the acetyl coenzyme A, which is ketogenic in nature. So it is apparent from the metabolic or catabolic pathway of this tryptophan that it is both glucogenic as well as ketogenic. So this is the more clear summary of the tryptophan metabolism right from 3-hydroxykynurenine from this step here. So this 3-hydroxykynurenine can convert up into the xanthionurate normally, which can be excreted in the urine. But if there is a deficiency of B6, so huge quantities, because there is a diversion to this pathway, and this lead to the huge quantities of release of xanthionurenate through the urine. So normally 3-hydroxykynurenine in the presence of kynurenase enzyme converts up into 3-hydroxyantranilate, which can lead to the important products via quinolinate, that is niacin. Niacin is a vitamin B3. So NAD and NADP are two important active forms of vitamin B3 or niacin. Then, on the other hand, this 3-hydroxyantranilate, after several steps, converts up into the acetyl coenzyme A. So, here in this pathway, alanine is also generated, which is glucogenic in nature. So, this is the summary of tryptophan pathway. So, one saying is there that for every 60 milligrams of tryptophan, 1 milligram equivalent of niacin can be generated. So, in vivo synthesis of niacin is definitely a sacrifice of huge quantities of tryptophan, means 60 milligrams equivalent are going to be converted into only 1 gram milli equivalents, 1 milligram equivalents of niacin. Move on to the next pathway, that is the serotonin pathway. In the serotonin pathway, the L-tryptophan is converted up into the 5-hydroxytryptophan via the tryptophan hydroxylase enzyme, which requires the tetrahydrobiopterin and molecular oxygen and NADPH. Then 5-hydroxytryptamine to 5-hydroxytryptamine with the loss of carbon dioxide through decarboxylation reaction. Further, this 5-hydroxytryptamine, that is 5-HT, that is also known as serotonin. So one specialized product, that is a neuroexcitatory neurotransmitter, is being formed here. Then 5-hydroxytryptamine converted up into the 5-hydroxyindole acetic acid via the MAO pathway, that is the monoamino oxidase enzyme, and which can be excreted in the urine. So MAO are very, very important enzyme system, and there you have seen in the previous lecture on tyrosine metabolism, that is, it is also responsible for conversion of epinephrine to metanephrine, and metanephrine basically convert up into vinyl mandelic acid in the presence of MAO. So further, this serotonin can be converted up into the melatonin via consequent acetylation and further methylation reaction. So first step there is a acetylation reaction via acetyl transferase enzyme, acetyl coenzyme A as a donor of acetyl group, which converts up into coenzyme A. Then this N-acetyl serotonin further methylated with the help of SAM, that is the S-adenosyl methionine, and on being methylated, it converted up into the N-acetyl 5-methoxy serotonin, that is the melatonin, which is an important hormone. The enzyme is methyltransferase. So in various reactions, metabolic reactions of creatine and tyrosine, also we have seen the important role of acetylinosyl methionine, and which act as an important donor of methyl group. So serotonin or the 5-hydroxytryptamine is a neurotransmitter. 1% of tryptophan is converted up into the serotonin. So synthesized from the tryptophan in the neurons, in the pineal glands, and in the intestinal argentophin cells. In mammals, the largest amount of serotonin is synthesized in the intestinal cells. Platelets also contains the high contents of serotonin. Then melatonin is a neurotransmitter as well as an important hormone, and synthesis and secretion happens in the pineal gland and well controlled by the light. Biologically important compounds derived from the tryptophan. So it is important for a precursor for the synthesis of neurotransmitters serotonin, melatonin, vitamin, niacin, that is V3. We have seen it previously. Then clinical significance of serotonin. Serotonin is involved in variety of behavior patterns, including the sleep, perception of pain, social behavior, in schizophrenia and mental depression. So in deficiency of this serotonin, there is a huge mental depression because it is a neuroexcitatory neurotransmitter. Functions of serotonin: it's a powerful vasoconstriction and stimulator of smooth muscle contraction in bronchioles and arterioles. Serotonin is an excitatory neurotransmitter and stimulates the cerebral activity. It plays an important role in the depression. Uh, in its deficiency, there is a depression. Serotonin controls the behavior patterns, including the sleep, body temperature, and blood pressure. Beside, it stimulates intestinal release of gastrointestinal peptide hormone. It controls the peristalsis. This is how has an important role in digestion. Then serotonin serves as a precursor for melatonin in the pineal gland. You have seen the steps of melatonin synthesis. Carcinoid tumor or argentaphenomas. It is a tumor of serotonin secretion. Carcinoid tumor developed from the argentophin cells of the gastrointestinal tract. Normally, 1% of tryptophan is utilized for the formation of serotonin, but in the carcinoid tumor, it is the 60% of tryptophan which is converting to serotonin. So it is completely diverted to the synthesis of serotonin production, and this leads to a lot of changes in the body. So the metabolism of tryptophan and decreased synthesis of NAD and NADP is there, and it's developed the symptoms of pellagra and negative nitrogen balance. Then clinical symptoms of a carcinoid syndrome are associated with prominent flushing, bronchial constriction, diarrhea, cardiac lesions, often associated with heart failure. Individuals have a high increase in urinary excretion of 5-indole hydroxyindole acetic acid. Measurement can be done by urinary testing of 5-hydroxyindole acetic acid concentration, which is increased up to 500 milligrams per day in case of carcinoid syndrome, right from the normal value of which is less than 5 milligrams per day. Then functions of melatonin: it is synthesized in the pineal gland from the serotonin. So synthesis of melatonin is regulated by circadian rhythm or diurnal variations, sleep-wake process. It is an inhibitor of melanocyte stimulating hormone, and adenocorticotropic hormone has an inhibitory effect on the ovarian function, so may be involved in regulating the reproductive functions as well. So move on to the metabolic disorders of tryptophan, that is the Hartnupp's disease. It is an inherited disorder of tryptophan metabolism. The name Hartnup disease it has derived due to it was first detected in the family of Hartnup. The defect lies in absorption and transportation of tryptophan and other neutral amino acid from intestine, renal tubule, and brain. that leads to low plasma level of tryptophan and increased urinary excretion. Then unabsorbed tryptophan in the intestine is metabolized by intestinal bacteria to indole acetic acid, indole pyruvic acid, which is subsequently excreted in the urine. And the tryptophan deficiency leads to the synthesis of vitamin niacin and serotonin adversely. And this leads to development of pellagra-like symptoms like diarrhea, dementia, and dermatitis. These are the three prominent features of pellagra associated with neurological symptoms, neuropsychiatric symptoms, as it is a neuroexcitatory hormone. So there is an amino aciduria due to the failure of transport of amino acids from the kidney, increased urinary loss of alanine, serine, threonine, asparagine, glutamine, leucine, isoleucine, phenylalanine, tyrosine, tryptophan, histidine, glycine, and citrulline. And it is an autosomal recessive disease. The net effect always lies in the excretion of these amino acids. So, diagnosis can be done on the basis of urine analysis, then detection of elevated amino acids via chromatography technique, and then urinary excretion of indole acetic acid and indole pyruvic acid and other derivative can be detected by the Ober-Mayer test, which is very important test for the diagnosis of indole ring compounds. Then molecular genetic testing is another important and cost-effective method to confirm the diagnosis of Hartnupp's disease. Therapies, as far as therapy is concerned, the oral nicotinic acid supplementation can give, and that leads to instant release by adequately synthesizing the NAD in the patient with Hartnupp's disease. But the amino acid urea cannot be checked by this treatment.